Hello, everyone, and welcome to today's webinar. I'm Melissa Romero, Forensic Science Consultant and CNE and Media Group. I will be moderating today's event, Solving the Complexities of T-Cell Engager Development, Pharmacology, PK, and Bioanalysis. This educational webinar is sponsored by WuXi AppTec. WuXi AppTec is a trusted partner and contributor to the pharmaceutical and life sciences industries, providing R&D and manufacturing services that help advance healthcare innovation. With operations across Asia, Europe, and North America, they offer integrated end-to-end services through their unique CRDMO, which is Contract Research, Development, and Manufacturing Organization platform. They are privileged to work alongside nearly 6,000 partners across 30 plus countries, supporting their efforts to bring breakthrough treatments to patients. Guided by their vision that every drug can be made and every disease can be treated, they are committed to advancing breakthroughs for patients, one collaboration at a time. CNN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the help widget at the top right corner of the screen or type your query into the Q&A box. If you are disconnected during the webcast at any time, please log in again according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. This webcast also allows certification of participation. To receive your certificate, please click on the certificate icon below your Q&A box to download your copy after watching the webinar. Please note that CNN does not endorse any company products or services that may be mentioned in the webinars, and each webinar will be archived at CNN online after the live webcast. During the presentation, we will be hearing from Jinshuan Yang and Dr. Xin Ning. Jinshuan is Assistant Director of the In Vivo Pharmacology Unit at WuXi Biology. He is a leader of the oncology platform at WuXi AppTec with a strong background in cancer pharmacology and preclinical drug development. Jinshuan earned his master's degree from the University of Washington and worked at Alta Sciences Preclinical Seattle for 4 years before joining WuXi AppTec. His work, characterized by the use of a wide range of sophisticated in vivo models, features a focus on higher translational value approaches. Additionally, Jinshuan possesses significant capabilities in preclinical formulation development, having established a platform of formulation screening for small molecules and PROTACs to accelerate clients' in vivo pharmacology studies. Dr. Xin Ning is Senior Scientist in the Bioanalytical Services of Lab Testing Division at WuXi AppTec. He received his BS from East China Normal University and his PhD in analytical chemistry also from East China Normal University. This included a 1-year PhD exchange experience at the University of Texas at Austin. Dr. Ning majored in bioanalytical chemistry and biosensors based in electrochemistry during his PhD period. He joined WuXi AppTec in 2021 and has served as an analyst, followed by principal investigator and project manager. Xin is now a core member of the business translation team within Bioanalytical Services. His work focuses on the clinical bioanalytical testing of various types of drugs, including bispecific antibody drugs. And let's get started. I will now turn it over to Jinshuan.
Good morning, everyone. I'm Jinshuan from In Vivo Pharmacology Unit at the WuXi AppTec. Today, me and Dr. Ning from our lab testing division will present our TCE platform with a powerful and comprehensive strategy to evaluate its pharmacology and efficacy in preclinical stage, as well as bioanalytical solutions to overcome its complexities in clinical samples. Thank you again for attending the webinar today, and now let's get the topic started. Cancer remains one of the most persistent challenges for human beings, driving an urgent need for more effective and targeted therapies. While traditional treatments like chemotherapy or radiation have saved countless lives, the field has been revolutionized by harnessing the power of the immune system itself, the immunotherapy. Among these groundbreaking approaches are T-cell engagers or TCEs. These are the engineered antibodies designed to act as a molecular bridge, physically linking a patient's own cytotoxic T cells directly to cancer cells. This forced approach activates the T cell, bypassing the need for antigen presenting and co-stimulation, leading to potent tumor cell killing. The power of this mechanism is already validated clinically, with several drugs approved for leukemias or multiple myeloma. This is the workflow of the preclinical TCE evaluation. The journey begins with TCE generation, where bispecific molecules are designed and constructed, typically targeting a tumor-associated antigen and a CD3 on T cells, using protein engineering to optimize format, affinity, and specificity. These candidates then undergo high-throughput screening to rapidly assess their ability to bind both targets and induce early T-cell activation signals in simplified assays. Promising these advance to in vitro functional testing using primary human T-cell co-cultured with target tumor cells to measure potent cytotoxicity against the antigen-positive cells and assess cytokine release. Finally, top-performing molecules proceed to in vivo pharmacology studies in humanized mouse models. These studies confirm anti-tumor efficacy, establish the PK and the dose-response relationship, and provide the initial in vivo safety assessment, and ultimately identifying the most promising candidate for toxicology and the clinical development. First, I'd like to introduce our work on generating and validating a novel TCE targeting DLL3 and CD3. We designed an IgG-based bispecific antibody using the nopes-in-host strategy to ensure correct heavy chain pairing. Some characterization was essential to confirm the integrity and purity of the molecule. SDS page analysis was performed to confirm the successful assembly and the purity. The expected molecular weights was verified by mass analysis, and the SEC analysis demonstrated a predominant peak of monomer with low aggregation. Also, the peptide mapping confirming the correct amino acid sequence and coverage. With a well-characterized molecule in hand, we proceeded to assess its functional binding activity. Here we use Jurkat reporter cells and a DLL3 expressing tumor cells to evaluate the T-cell binding to both CD3 and the tumor antigen. We employ the facts to quantify the binding affinity. The results demonstrate a successful generation of a high-quality TCE molecule with the desired binding properties to both DLL3 and CD3. Knowing the level of target expression on our test cells allow us to understand the binding affinity results. Here we provide such essential data by quantitative facts. Here is an example of screening a surface target expression in multiple cell lines. To further characterize the functional activity of TCE, we have various assays established to evaluate the T cell activation in vitro. Here in the top case, by using flow cytometry, we observe a dose-dependent increase in the surface expression of both CD25 and CD69. The bottom case is a cytokine release test by ELISA. We quantified the secretion of key effector cytokines, interferon gamma, and TNF alpha in the supernatant post-co-culture of 24 hours. We can also employ multiple platforms such as MSD and Luminex assays to provide a more comprehensive picture of T-cell activation by TCE. After confirming T-cell activation induced by TCE, we move forward to an in vitro T-cell killing assays. These experiments evaluate the ability of T cells engaged by TCE to kill target cells expressing tumor antigen. The top case presents our initial screening using a calcium release assay. This method measures the cytotoxicity by detecting the release of a fluorescent dye from preloaded target cells upon lysis. Here we optimize the ET ratio using two distinct target cell models, both co-cultured with human PBMC serving as a source of T cells. This screening was crucial for identifying the optimal ET ratios that would provide a robust signal window for subsequent detailed killing assays. The cytotoxic activity of the TCE in a T-cell mediated killing assay can be quantified by flow cytometry as well, like the bottom case. To gain deeper insights into the kinetics and the specificity of T-cell mediated killing assay induced by TCE, we established a novel reporter system. Target tumor cells engineered to express both TD tomato, a bright red fluorescent protein, and the nanoluciferase. The constitutive expression of TD tomato allow us to easily identify and track the target tumor cell population throughout the co-culture period using IncuCyte. We can directly see the loss of TD tomato positive cells in the presence of T cells and the TCE over time. The nanoluciferase will benefit further tracking in the in vivo test. Here is an example of using DAO reporter MCF7, an antigen-negative cell, and the U2OS, an antigen-positive cell. We observed an obvious tumor cell killing in the antigen-positive cell co-culturing with CD3-positive effector cells post the treatment of the bispecific antibody. When moving forward to the in vivo test of TCE, the immuno-oncology models are necessary. The humanized model is usually preferred because most TCEs target human-associated tumor antigen and don't have cross-reaction with murine cells. Knock-in humanized models, where mice are genetically engineered to express specific human tumor antigens or immune molecules like cytokines or checkpoint receptors within an intact murine immune system, offer a powerful platform. These models allow us to evaluate the interaction between human-targeted therapies and a functional syngeneic immune system, providing valuable insights into the pharmacology of antigens like checkpoint inhibitors. Complementing these are human immune system models, generated by engrafting human HSC or human PBMC into immunodeficient mice. In such models, human cancer cells or patient-derived tumor samples can be implanted for xenograft model development. At WuXi AppTec, we have established over 90 PBMC or HSC humanized models in-house, covering 27 cancer types. We've also validated many targets like CD19, CD3, and the EGFR CD3 using positive controls in these humanized models. Although both PBMC and HSC xenograft model offers human immune system, the human HSC model develops a more complete and durable human immune cells over time, enabling the studies of human immune development and the long-term responses against the human tumor xenografts. Alternatively, the human PBMC model offers a rapid transient reconstitution of human T cell mainly, making it highly useful for short-term efficacy and safety assessment of T cell redirecting therapies like TCE or CAR-Ts, although the GVHD remains a limitation. Let's begin with our first case study, evaluating an anti-Claudin 18.2 CD3 TCE candidates. For this case, we employed a human Claudin 18.2 MC38 in human CD3 EDG humanized mouse model. The human CD3 EDG humanized mouse strain was developed by replacing the mouse CD3 EDG genes with their human counterparts, resulting in expression of a human CD3 EDG complex while knocking out the endogenous mouse genes. For the cell line, the human Claudin 18.2 coding sequence was inserted into the MC38 cells, resulting in high expression on the surface. So this model features transgenic expression of both human CD3, which enables T-cell engagement, and the human Claudin 18.2 on MC38 tumor cells. This allows us to study the TCE activity against a solid tumor expressing the target antigen in an environment with functional human T cells. And as you can see from the tumor growth curves, the administration of the anti-Claudin 18.2 CD3 TCE resulted in a significant tumor growth inhibition comparing to the control group. To understand the mechanism behind the efficacy, we performed the TIL analysis and observed an increase in T cell infiltrating within the tumor microenvironment, especially the CD8 positive T cells. We also tested this anti-Claudin 18.2 CD3 TCE in a different humanized mouse model. Here in a second case, we used a more complex human immune system model by engrafting human HSC into the NOG-EXL mouse who express human GM-CSF and interleukin-3. As for the cancer cells, we used human pancreatic cancer cells, MIAPACA2, and it was also engineered to express human Claudin 18.2. Upon a parallel injection of cancer cells and human HSC to such immunodeficient mice, tumors were developed and a diverse human immune system, including myeloid lineages, were well reconstructed. The tumor growth curves demonstrated that the TCE maintained its potent anti-tumor efficacy against the Claudin 18.2 expressing pancreatic cancer within this more physiologically relevant immune context. And the TIL analysis confirmed the significant T cell infiltration into the pancreatic tumors and its activation. Our last case study shifts the focus to hematology malignance target, the CD19. We evaluated an anti-CD19 CD3 TCE candidate using the ibrutinib-resistant patient-derived lymphoma model in a human PBMC humanized mice. First of all, the ibrutinib-resistant model was established by a long-term treatment of ibrutinib in vivo until a resistant phenotype is confirmed in this patient-derived B-cell lymphoma model. To overcome this acquired resistance post-treatment, we would apply an immunotherapy by an anti-CD19 CD3 TCE, but its humanized model needs to be established first. This time, we engrafted the mice with such patient-derived ibrutinib-resistant B-cell lymphoma tumors, followed by human PBMC injection to provide the human immune effector cells. The tumor growth curves clearly show that the anti-CD19 CD3 TCE effectively control the growth of the resistant lymphoma, and the TIL analysis also revealed the T cell tracking to the tumor site. The last but also the most critical component of our TCE preclinical platform is its robust PD marker detection capability, especially designed to elucidate the complex interplay within the tumor microenvironment and the resulting immune context. Overall speaking, our PD marker detection team offers multiple layers of sample analysis from gene-based to protein-based, cellular-based, and tissue-based analysis. For TCE drugs, we employ advanced multiplexed assays, including spatial profiling techniques to quantitatively measure key biomarkers and its activities. such as cytokine release by MSD or Luminex and the TL analysis by FACS. This integrated approach provides a high-resolution view of tumor microenvironment modulation, enabling us to assess changes in immune cell infiltration, functional status, and immunosuppressive networks. By comprehensively evaluating these PD responses within the dynamic tumor microenvironment landscape, we provide insights into TCE mechanism of action, efficacy, and potential resistance mechanisms or toxicity early in development. At WuXi AppTec, we have three pillars, namely WuXi biology, WuXi chemistry, and WuXi testing. WuXi biology offers drug discovery and early development in the field of biology and diseases without specific modality limitations. WuXi testing provides DMPK toxicology and bioanalytical services. WuXi chemistry specializes in the chemical synthesis, design, and development, primarily focusing on small molecules, but increasingly including complex molecules and new modalities like peptides. These three divisions work together to deliver our end-to-end solutions to our clients. In WuXi Biology, we provide comprehensive discovery and translational services from target to hit, hit to lead, lead to PCC, and to preclinical pharmacology evaluation in disease models. We cover various modalities like ADCs, bispecific antibodies, oligos, peptides, bifunctional molecules, and so on. We've also developed thousands of assays and animal models covering different diseases like oncology, immunology, neurodisease, metabolic disease, and so on. We work as an integrated team by bridging different functional areas in a shared system and a simplified workflow. Okay, now let's welcome Dr. Ning, my colleague from WuXi Testing, to give us a talk about the clinical bioanalysis of T-cell engager.
All right, thank you, Director Yan, for the presentation. So this is Xin from the Department of Bioanalytical Services at WuXi AppTec. So following Director Yan's speech, we are going to have a deep dive into bioanalytical considerations on T-cell engager across PK, immunogenicity, and biomarker analysis. So firstly, let me give you a brief introduction of the Department of Bioanalytical Services at WuXi AppTec. So the Bioanalytical Services at WuXi AppTec, abbreviated as BAS, has leading integrated laboratory operation in both China and the United States, with more than 20 years lab operations in China and America. The headquarters of BAS is in Shanghai, with sites in Suzhou and Nantong in China and New Jersey in America. The Shanghai site mainly focuses on clinical study, and sites in Suzhou and Nantong are working on preclinical studies. The New Jersey site now focuses on central lab services, adhering GLP and CAP systems. Our lab has long-term lab operations and experienced specialists in different fields, with more than 600 employees. Among them, nearly 7% have master or PhD degree. So coming back to our topic, firstly, in the perspective of clinical bioanalysis of protein therapeutics, the scope encompasses the comprehensive analysis of drug molecules in biological matrices to determine their safety, efficiency, and dosage during clinical trials. It involves the quantitative measurement of the drug, the monitoring of biomarkers, and the assessment of immunogenicity. So key areas of clinical bioanalytical scope include PK, immunogenicity, and biomarker. PK studies quantify drug levels and assess drug exposure. Immunogenicity studies include screening, confirmatory, and titer assay, together with neutralizing antibody assay if needed. PD and biomarker studies mainly focuses on biomarker analysis and the evaluation of the correlation with PK and immunogenicity. To ensure the quality of the clinical bioanalytical studies, together with keeping the clinical bioanalytical process in a smooth way, clinical bioanalytical guidelines and regulations provide a framework for the validation of bioanalytical methods used in clinical trials. Key requirements include method validation with specific acceptance criteria for accuracy and precision, ensuring the reliability and consistency of data across all phases of drug development. Furthermore, documentation is crucial, requiring the maintenance of records for all steps of the process, from sample handling to analysis, to ensure data integrity and allow for full reconstruction of the study. Primary guidelines and regulations are mainly from ICH, like ICH M10, US FDA, EMA, or WHO. Besides, NMPA, which is China FDA, also has guidelines and regulations for clinical bioanalysis. For biomarkers, the fit-for-purpose principle is now applied into the clinical biomarker bioanalysis. The principle of fit-for-purpose bioanalytical guidelines means that the level of validation for analysis should be appropriate for the intended use of the study. The required region of the validation depends on the role of the data generated by the assay in the drug development process. The FDA guidance on bioanalytical method validation for biomarkers, published in 2025, specifically addresses the validation of bioanalytical methods for biomarkers, which were excluded for the ICH-M10 guidance. It endorses a fit-for-purpose approach for biomarker assays, and the FFP approach tailors the validation strategy based on the intended use of the data, which often corresponds to different stages of drug development. And unlike PK assays, which have a single well-defined purpose, biomarker assays can serve many different purposes throughout drug development. The fit-for-purpose validation of a biomarker method is driven by its context of use and scientific justification. So note that with the implementation of the ICH-M10 guidelines, the FDA's 2025 biomarker guidance now aligns with the ICH framework, creating a clearer, more consistent global approach for bioanalytical method validation. Regulators encourage sponsors to use ICH-M10 as a starting point, tailoring the validation plan to the specific concept of use of each biomarker. So coming to the TCEs, PK, immunogenicity, and PD are within the scope of clinical bioanalysis. The PK section includes PK and TK analysis, while TK may mainly be evaluated in the immunogenicity and PD are within the scope of clinical bioanalysis. The PK section includes PK and TK analysis, while TK may mainly be evaluated in the preclinical stage. The immunogenicity section includes AD and NAb, beyond standard PK/AD assessment for bispecific antibodies. T-cell engagers often induce severe cytokine release syndrome and immune effector cell-associated neurotoxicity strategy, which normally co-occur. So to establish robust PK/PD models for TCE drugs, monitoring T-cell or other major immune substance counts, T-cell activation models for TCE drugs, monitoring T-cell or other major immune substance counts, T-cell activation, proliferation, and target cells numbers is essential. So in addition, concurrent monitoring of B cells, NK cells, and monocytes. Concurrent monitoring of B cells, NK cells, and monocytes is commonly conducted to assess drug-mediated cytolysis. These assays normally rely on flow cytometry-based immunophenotyping techniques. So regarding the bioanalytical challenges, sensitivity and specificity are common focuses for most of the PK/AD NAb and PD assays. Besides available critical regions in PK assays, region labeling in AD assays, or gauge setting in flow cytometry are also challenges for clinical bioanalysis.
To have a deep sight on the challenges of TCE clinical bioanalysis, examples will be raised in the coming presentation. So here presenting the TCE clinical bioanalytical assays regarding PK and immunogenicity analysis. The goal of PK bioanalytical assays regarding PK and immunogenicity analysis. The goal of PK analysis is to measure the amount of the drug in the patient system over time to understand its absorption, distribution, metabolism, and etc. The PK analysis will be total TCE measuring all forms of the TCE in the bloodstream. Free TCEs measuring only the unbound forms of the TCE in the bloodstream. Free TCEs measuring only the unbounded active drug available to engage T cells and tumor cells and specific complexes. So the assay platforms are mostly ELISA or MSD for immunogenicity assessment. Since TCEs are proteins, they can trigger an anti-drug antibody or MSD for immunogenicity assessment. Since TCEs are proteins, they can trigger an anti-drug antibody, which is ADA response in patients, an anti-drug antibody, which is ADA response in patients, potentially affecting both safety and efficiency. So 3 tiers of assays: screening, confirmatory, and titer assays make up the whole ADE assay. So the assay platforms are mostly MSD, make up the whole ADE assay. So the assay platforms are mostly MSD. So for assays of detecting neutralization antibodies among the ADA, cell-based assays preferred as it can better align with the TCE's mechanism of action, thus analyzing the neutralization antibodies in a more accurate way. So for some different situations, the ligand binding assay does not work in the PK assay. Hybrid LC-MS platform can also be an option for the setup of PK assay. Its mechanism of action, thus analyzing the neutralization antibodies in a more accurate way. So for some different situations, the ligand binding assay does not work in the PK assay. Hybrid LC-MS platform can also be an option for the setup of PK assay. So hybrid LC-MS procedures combine the selectivity of immune affinity capture using antibodies and with the analytical power of LC-MS mass. The process typically involves capturing the target analyte at first, and then digesting the captured protein into smaller surrogate peptides, then separating these peptides with LC, and finally analyzing them with MS mass to select, fragment, and detect the specific peptides. So this hybrid approach is highly effective for quantifying large molecules like protein therapeutics in complex biological samples. Also, this approach combines the selectivity of ligand binding with the sensitivity of mass spectroscopy. And it can be used to specifically quantify different forms of the drug and its biotransformation products. PD assays measure the drug's biological effects on the body. For TCEs, this centers on evaluating T cell activation and tumor cell killing. Cytokine release syndrome, abbreviated as CRS, is a common side effect of TCEs. CRS is caused by the release of pro-inflammatory cytokines like IL-6, TNF-alpha, and interferon-gamma. Measuring these cytokines is essential for assessing and managing toxicity. Platforms like ELISA, MSD, Luminex, Simova, or Ella are used for cytokine analysis. So the truth of these platforms depends on the demand of sensitivity. Regarding the immunophenotyping, flow cytometry is used to analyze immunocells from blood or tissue biopsies. This can track the expansion and activation of T cells and monitor their migration to the tumor site. Also, the receptor occupancy can also be evaluated using flow cytometry platform. Also, pathology is a pathway of evaluating the drug effect. This can be used as one of the indicators for patient screening and can stratify patients. And besides, immune expression before and after medication can also be evaluated.
Moving forward, let me present some clinical bioanalytical cases of TCEs. Issues or challenges will be discussed here via some cases. Firstly, let us have a view on the issue of target interference in the PK assay. So for the PK assay, a soluble target in the human matrix may interfere the performance of the assay by binding with the drug, so that preventing the drug from binding with the reagents, especially when using the target as the critical reagents like coating or detection. So to avoid this, the first thing coming into our mind is to optimize the assay format, which means using the specific antibody against the drug to replace the target as the critical reagents. So as for the pre-binding drug target complex, if any, our aim is to break the drug target binding to release the drug so that the released drug can be captured by the coating reagents. So assay dissociation is one way to break the binding between the drug and the target. But one thing needs to be noticed is that this assay dissociation way is not available for the drugs that are sensitive to different pHs. The structure of these kinds of drugs may be altered when the pH is changed. Another way of avoiding the target interference in the PK assay is that the free targets in the samples can be depleted in the sample pretreatment procedure. For example, the anti-target antibody mobilized beads can be used to enrich the target in the samples first, then remove the base complex from the samples so that the target is moved together out of the samples. And furthermore, if the ligand binding assay cannot work, LC-MS mass assay is also a choice for the PK analysis. The advantage of LC-MS mass assay comparing with the LBA assay is that the LC-MS mass assay may be a reagent-free assay. So digested specific fragments are detected via LC-MS mass platform with no impact by the target.
So this is one case to show that different assay formats have direct impact on the assay performance. For ADA assay, usually the bridging format is used. Therefore, the bridging format is firstly used while the results shows that the signal is low, even though the PC concentration reaches to 16,500 nanogram per milliliter. And the difference of the signal under the same concentration in monkey serum and PBS buffer is quite different, indicating that this format is not robust or stable enough. So when changing this bridging format into an indirect ELISA format by changing the detection reagent from the ruthenium labeled drug into the ruthenium labeled anti-human IgG antibody, things get better as there is an obvious increasing trend of signal when the PC concentration goes up. For comparison, when the new format is in use, the signal at 2000 nanogram per milliliter is even higher than the signal at 16,500 nanogram per milliliter when using the old format. Besides, the precision of this new format looks good as the difference of the signal under same concentration is small. So another point needed to be noticed in the setup of an AD assay is that the drug tolerance needs to be treated carefully. And the drug tolerance is evaluated based on the dose level, and sometimes the drug tolerance level may fail to meet criteria. In this case, the drug tolerance level needs to be evaluated in conjunction with the PK results. If not okay, additional validation of the increased drug tolerance level based on the original ADE assay is needed to cover the higher dose level. And if the additional validation is not successful, the ADE assay needs to be upgraded or a new ADE assay needs to be set up to obtain a higher drug tolerance level. So technically, to improve drug tolerance in ADE assay, a starting step is to increase the incubation time in the assay using a homogeneous bridging format. If that does not achieve the required drug tolerance, sample pretreatment can be used. Sample pretreatment often uses assay to break up the drug ADA complexes. isolating the ADAs from the sample and then running them in the bridging assay. So here's one case that using flow cytometry to perform NAB assay, and tumor cells are firstly stained by cytoplasmic dye and set in the plate. After mixing with the immune cells, samples will be added into the plate wells and then delivered onto the flow cytometry to perform the analysis. So in the existence of neutralizing antibody, the drug is combined by the NAB so that the tumor cells cannot be killed by the immunocytes. With the increased concentration of the NAB, the cytotoxicity percentage goes lower, hence illustrate the trend of the increased NAB level. All right, so that will be all for the presentation, and thank you all for listening. And if there are any questions regarding this presentation, now we have time to have a discussion. So your questions are welcome now.